Okay, so yeah, happy that everyone can join this workshop because I think that is the second workshop of the EBP-IT Informatics Standard Subcommittee. The last one is held 2 years ago. So I think that there is much updates in this area of the IT and informatics in the genomics research. So the first one I quickly will make some introduction about the agenda of these 2 days. There is 4 hours of this workshop. And today we have a section one, and the section one, the topics is about the data management best practice and the challenges. And in this section, we'll cover essential subtopics of genomics data management and including the best practice of the I5K standards by the I5K standard working group, and the introduction of the bioheaders about the data accessibility and all the other design about the bioheaders. There are some, maybe they have some tools and the insights from the NCBI team. We'll make some discussion about medical data repository concepts and also the management principles. And this section two is about the genomic. Okay, just enlarge screen. Yeah, the section two is about the genomics informatic tools and database because the EBP, the IT, and the informatics subcommittee will focus much on the informatics things and just like the tools and database on some other platforms. So in this section will be our main part. So we discuss some key resources or platforms or systems. The first one is maybe someone knows the genomes of the trees, the goat. It's a centralized search engines to get the information about the genomes, and also we will make some introduction on the DCS Cloud. It's a new platform developed by BGI teams, and also the Galaxy platforms. And we hope that because many people are quite familiar with the Galaxy, and we hope that we can for this section that everyone can get more information about all is a roadmap or any new design in this system. And the third one is also about the genomics informatic tools and databases. And we will have made some discuss about the standards of writing the pipelines, the three double lines, example. And also Emilio will make some introduction of the BGP portal. And Dr. Terence will make some introduction about the NCBI toolkit and to how to enhance some use of the genomics data. And the fourth one will still make a couple of new discussions about also some biodiversity portals of the EBI, and also introduction of the whole genome duplications, some technical things, how to analysis the genomics data. And also Jerry from the National GeneBank, we right now have some concepts of the develop a system we call the Biomedical, and he will make some brief introduction about this part. So from today and this week Thursday we have 4 sections, and today we have section 1 and section 2. Yeah. So the second part we still want to make some introduction about the EBP and our standards. Yeah, the first one is a screenshot from the Sibeli's the Gold homepage, and it's quite useful tools. You can get to know what is the progress of the EBP projects, and maybe other projects is also work well, and most of the data, the metadata we get from ISDC, because there is a EBP umbrella project right now in NCBI. So it's quite useful. You can get the useful informations, what kinds of species already sequenced, and what you need to do, or what kinds of species you need to sequence in the future, and also the progress of all the projects in EBP. And also the EBP-IT and informatics standards. And right now we already have visions and the latest vision updates last year. And I hope that after this workshop we can compile all these reports and all these topics and make them as a best practice to these EBP-IT and informatics standards. And because you may know that IT and informatics it's a little different, right? With the sample collections or some sample management system or some sequencing tech or some data analysis pipelines or some or even some other kinds of details or some work you do in your research or genomics research. It's a little far away from them because sometimes not much teams they focus on these parts. So I hope that after this workshop, all these can take it as a best practice and we can move these and genomics standards in the EBP forward. So I also put because I like sometimes because I painting and also take photograph every day. So there is Stephen Shower. He's an American photographer, and he has an art book called Uncommon Places. And this picture is one of his favorite pictures. His work, and there is some new concept from his photograph, but I'm not sure that everyone like photography. But has a words that a sentence, a photography has ages, and the word does not. So I think that it's the same set to our genomics research. So I hope that from this workshop, everyone can get what you can get. Okay, so so maybe we just start, and I just stop the sharing. And the first speaker is the Professor Marc from the University of Buffalo. And his research is focused on the genetic and regulatory that maybe some cells facts that during the development. And his teams also focus on the bioinformatics database and had developed maintains a database called Red Flies. It also is recognized by many internationally right now. And also the I5K is a global genomic community. And it's a community that ensures that all of the insights or some genomics research and the information need to in the future can perform or can be used across different inside scientists. Yeah. So Professor Marc, maybe you can share your presentation.
Yeah.
Thank you.
See if this loads up. Okay, how's that? Oh, thank you, and good morning or afternoon or evening, I guess, depending on where everybody is joining in from. It's morning for us. Yeah, I'm going to speak today as a representative of the I5K Standards Working Group. And what I'd really like to do is deliver sort of an interim report on what we've been working on over the past year. So this is very much a work in progress, and we're very much interested in feedback from other members of the group. I'll try to leave a little bit of time at the end, or people can contact us, of course. Here's just the members of our working group. Like I say, I'm really just representing them. This is a very much a group effort. And I thought I'd just start with a brief introduction to the I5K group. The I5K is a community that was formed about in 2011, 2012 with the idea of facilitating the genome sequencing of initially 5,000 different arthropods, primarily insects. In recent years, of course, we've come to that goal. We now have over 6,000 arthropods that have been sequenced, including over 1,500 chromosome-level assemblies. And so about 2 years ago, as we started reaching that goal, we started talking to our members, and this just sort of shows where our representatives come from. We are well distributed around the world. We've recently done some reach out in Africa, and we hope to have more members participating in I5K coming from there as well. But we've reached out to our members about 2 years ago to start a revisioning process and said, well, now that we're sort of nearing this initial goal of this many arthropod sequences, what should the I5K community be doing? And we formed this vision statement and mission statement. The idea is that the I5K is a community of arthropod researchers. We should ensure that these genomes are available to all researchers who need to use them, and that the information and the protocols and the methods and the standards that are needed to work with these data are available to the whole community. And to fulfill that vision, we sort of formed as our mission that we would promote the best practices for genome projects, support the generation, access, analysis, visualization, and understanding of genomic and post-genomic data, as well as present recommendations to those who give out funding to try to ensure that we have the support that we need to continue to pursue these goals. In order to fulfill this mission, the I5K formed a number of different steering committees, and today I'll really be talking to you about the standards working group subcommittee. And the idea of the standards group was to come up with guidance for genome projects and to publicize best practices and the existing standards for working for them, as well as to develop new standards as may be needed. This came directly from feedback from our I5K community, who clearly requested this type of guidance to work on their projects. And this came out of a series of sort of focus groups that we conducted about 2 years ago. And you can see, I won't go through the whole list here, but members of the I5K community, of the arthropod genomics community, really were looking for, could we make recommendations on specific techniques? What standards are out there? What are the best ways to deposit the data? Where should data be deposited, and sort of what formats should be used for that? So there was a lot of need from the community for guidance on these issues, and that's why we formed this working group to discuss standards. Initially, we decided that the place to start would be by just conducting a sort of gap analysis to say, well, what standards are out there? What standards are missing? What practices are in use? What practices, need to be better publicized and incentivized amongst our community overall, but in particular as that obviously pertains to arthropod genomics? So are there special considerations for arthropods that we need to have there? And to do that gap analysis, we basically followed the genome workflow that was laid out by the Earth Biogenome Project, right? And so we broke it down into all these different areas that were laid out by that group: funding and budgeting, sourcing and vouchering, sequencing, assembly, gene prediction, databasing, various downstream analyses, and just sort of brought a group together to start looking at what standards might exist there and what gaps there would be. So we met a little over a year ago, our first group, and we brought together the working group, plus we invited various domain experts in these areas who would have expertise to lend us. And the idea would be, to look, again, specifically what we need for arthropod genome projects, but also what may be needed in general there. So what are the existing standards and practices? What requires modification for our group? And what standards or practices are really lacking, either for everyone or for insects and arthropods in particular. So the first thing that we realized as we started doing this is there are actually a lot of standards in place. And a lot of this is thanks to all the excellent work that the EBP has done and that are publicized. And so, again, I'm not going to read through everything, but sort of you can see on the right of these slides as I go through them that there are a lot of existing standards that are linked to out there. So we had a very good starting point for this. And what we really did not want to do was reinvent the wheel and go back and rewrite standards that are already there. So we had very good starting points really for all of our areas: for sequence assembly, analyses, for gene prediction and databasing. Those all existed from the EBP. But we realized from this that there were sort of two types of gaps that we should be considering. One would be what we decided to call standards gaps and focus on. And the standards gaps are those places where the standard or metric that we need really doesn't exist yet, right? So either there wasn't an existing standard up there from EBP that we could rely on, or those standards didn't quite fit the arthropod genomics community, that there were maybe some differences there. And so these would be cases where new standards need to be developed, and we need to get to work and figure out what those standards should be. Okay? The other type of gap we realized is what we decided to call a practice gap. And what we mean by a practice gap is this is a place where those standards do exist. So the processes and metrics that we need are defined as a standard, but we're finding that they're not commonly or consistently used within our community. And so these are places where we don't necessarily need to develop a new standard, but where our community needs to be educated and or incentivized in order to adopt these standards and make use of these standards. And in fact, we found this to be much more prevalent than the lack of standards altogether, again, because so much work has already been done by EBP and other groups. What we also realized, although we were really focused on our community, a lot of the gaps we identified really weren't arthropod specific, but were sort of generic genome project issues and places where overall there weren't good standards, or those standards weren't being widely adopted, not just within our community, but in general. And so although we started with sort of a narrower focus, we think that that sort of broadened out as we've done this, and we think that the information that we end up with is going to be of broad use because it's going to affect much more than just the arthropod community. So I'm just going to quickly run through what some of these gaps that we identified were in our visioning process. Again, this is just an interim report. I'm not going to go into a lot of detail on this. This is just to give an idea of what we have. And we sort of outline these out, whether these are practice gaps or gaps in the standards themselves, or some combination of both, where there need to be new standards, but also they need to be more widely adopted. And so we started with the budgeting and funding category. And of course, there aren't necessarily direct standards, so to speak, for that. But we did recognize that in practice, some projects start out without really having a good sense of what resources need to be accrued and what needs to be there. And we thought there's room for guidance there for people who want to embark upon a genomics project. And of course, now I'm not talking about the big consortia, but I'm talking about smaller individual groups who might have a organism of interest that they want to do. And what we recognized was that if these resources weren't really in place and carefully accounted for in the beginning, a lot of these projects would peter out and never really reach a fruitful end. And you'd end up with some sequence, but not really a good genome. And so this is an area where we need some education and some guidance. The same for data management plans. There's some research that shows that when a good data management plan isn't put in place in the beginning, at the end of projects, a lot of time data end up not being easily accessible or widely accessible. And so again, these are places where there both should be a good sort of standard template that people could use, but also education about how to use that and do it. And the same with reusability. People need to understand where to publish the data and what sort of formats to use. In terms of assembly, there were gaps both in standards and practices that we recognized. One issue with standards, and this is something I think the community in general still needs to coalesce around, is what should replace things like the N50 measure. It's not a very useful measure in the days of chromosome scale assemblies and really long read sequences, where we expect the contigs to be really long and for there to be relatively few of them, right? So that becomes not such a good measure. But our feeling was that the community in general hasn't really coalesced upon good alternatives to that and what those should be. So that's an area where more work needs to be sort of with everybody in terms of standards. But there are also practice gaps. There are some metrics for some things like genome completion that can use K-mer-based methods and things like that to get good measures. But we find that often these aren't being used by people in their projects. And so that's a place where the practice needs to be improved. The same is true for assembly correctness. Curation, contamination. There are good standards and there are good methods to take care of contaminating sequences and sequences from cobionts, but not everyone is aware of these. Not everyone uses these, and you find these sequences creeping into assemblies and all. And so that's another place where I think the technology exists, but we need to make sure that it's being widely adopted by all users in the community. In terms of analysis, reproducibility is something that's very important, obviously. There are good workflow managers, and there are a lot of things being done in that area. But that's a space that, in our community, at least, in the arthropod community, it still needs to see wider adoption, and using standardized workflows and reproducible workflows that are out there that people can then make use of and reanalyze data and reproduce analyses. That needs to be improved upon. Annotation, again, gaps in both practices and standards that we noticed. There are good methods and standards out there for dealing with repeat annotation and gene annotation, but again, these aren't always adopted by all members of our community. Something near and dear to me is regulatory region annotation. And there, not only do we not have good standards for doing that, it's not clear what are the best ways to do regulatory annotation. It's also something that's often overlooked, and most genome annotations that get published really don't include much in terms of the regulatory genome and really only focus on the coding genome. Quality assessment here is a place where we think there are need for additional standards and additional work. So BUSCO and tools like that are often used. We find in our community, at least, that those are heavily weighted towards dominant model organisms such as Drosophila. And therefore, as we start getting into more distant arthropods and different members of the phylum, that those tools aren't really sufficient because they don't cover a lot of the specific genes that are there. And so we think there's room to find some replacements and other ways of looking at this that need to be thought about. And that goes for quality assessment too, as well as nomenclature. Again, in the arthropod space, heavily dominated by Drosophila. That doesn't bother me particularly. I'm a longtime Drosophilist, but it does lead to problems as we try to standardize names and functions and identities of genes and gene products, of course, that there needs to be good standardization, and so we can have good cross-referencing and so forth. Comparison with previous annotations is something we identified as a problem. So transferring annotations and knowing which annotations to refer to and how to refer to them, and how to report these. So there is a standard, obviously. GFF has a standard, but it's a loose standard. And there are a lot of different ways to do your annotations in a GFF file. And we find that those vary a lot from project to project. And I can tell you from my own experience that breaks a lot of software pipelines because things are listed differently in that final field of the GFF format. And they can be hard to parse and hard to follow. So there we need better standards, we think, and we need to publicize those and work for them to be more widely adopted. Databasing is a big problem. So there are a lot of databases out there, but some of them can be hard to find. Funding to maintain smaller community databases is an ongoing issue, and what happens is things then disappear or become less functional, and they're not always permanently archived in ways that lets someone access data once they sort of fade out of existence because of funding difficulties or for whatever reason. We need better standards for interoperability among these different databases. There are a lot of different frameworks and different identifiers that get used. If that would be more standardized, then we could cross-reference and interoperate across databases better. So there's room for standards there, and also practices. So there are certain standards and formats out there, and it would be useful if those were more widely adopted. There are some database hubs that can be used. The National Agriculture Library has the I5K workspace where people can deposit their insect genome projects, for instance. But again, not everyone does that. Not everyone takes advantage of some of these pre-existing formats and pre-existing software that's out there in order to be used. This is more of the same, really. So again, just better interoperability, both in practice and with standards for that, would be very helpful in terms not just of the gene annotation, but phenotype databases and gene functions as well. Okay, so that's kind of our interim analysis and sort of where some of the key gaps are. And there's other stuff in there. I'm not going to go into excessive detail in a talk like this. What are we doing with this? So in the short term, we're taking the following steps. One is we've been working on a manuscript describing some of those key gaps that I just outlined, where there are standards gaps, where there are practice gaps. And we hope to have that manuscript completed relatively soon, actually. And that should provide some talking points and some reference point to continue doing work in this area. We're also undertaking an analysis of genome reports and genome publications over the last year or so to sort of get actual data gauging the incorporation of those standards that are out there. So places where we feel there's a practice gap and that standards aren't really being well adopted, we'd like to gather some data to really see what the extent of that is and whether that's so or not. And we're also, as sort of an interim step, working on a manuscript describing sort of best practice recommendations for writing and submitting genome reports. So we have canvassed the journals that publish these type of supports to find out what sort of data that they require and in what sort of formats. And we'd like to compile that along with our recommendations of additional things that people should keep in mind when writing and submitting those reports. And that should help the field in general sort of stay up to those standards that we have at least and fill some of those practice gaps. In the longer term, we're interested in developing educational materials to advocate for or disseminate information about the existing standards, that is, address some of those practice gaps, right? Which again, a lot of the time is not because people are unwilling, but just because people are not knowledgeable about some of the standards that are out there or don't have the proper incentives to make use of those standards that are out there. And so I think by educating the community and perhaps working with some of the publishers that we can make a dent in some of that. The other long-term item that we need to do, obviously, is to form some teams to work to develop those standards that are lacking, that we've identified that are needed. And we hope to begin doing that in the future once we sort of finalize our gap analysis. And for that, we will almost certainly be reaching out to other members of the community, not just arthropod people, in the interest of developing good and proper standards in places where they're currently lacking. Okay, I always started a little bit late, so I'm going to try to wrap up a little bit early so we can keep on schedule here. I just want to thank the members both of our working group as well as all the participants in our initial gap analysis workshop.
Lead the workshop. Maybe job, right? Yeah.
I lost
Okay.
the Zoom there somehow.
Yeah.
Let's try that again. I don't know what happened. Okay, how's that? Are we back? I was just trying to thank everyone who participated here in our both the working group and the people in the initial process. And I apologize if I left anybody off of this process. But this input from members of the community has been absolutely vital in our being able to do this work. And I thank as well all the members of the I5K consortium who have participated and lent their expertise and has also lent their questions and what their gaps and what they need to know may be. And with that, I'd be happy to answer questions or take a little discussion if we have a few minutes, because we are interested in pushing this forward and knowing what people think about some of these gaps or recommendations to fix them. And obviously, if they're interested, people who are interested in working with us, please contact me later on, and we're happy to have people participate. So thanks.
Thank you. Thank you, Professor Marc. Yeah. So, yeah, debating. Okay. Anyone wants to raise your hands? Yeah. Okay. Yeah. Sorry. Yeah. So I found that Mark, your standards cover from the specimen, from the sequencing analysis, annotation, and also the database, or and even in the future the application. So because right now in our teams or maybe some other teams also focus on some images about the specimen. So so do you have any ideas about this part? The images of the some specimens or
Yeah, so
the vultures.
that's something that we're attuned to as part of the specimen and vouchering part. We haven't talked that much about that aspect yet. We are aware of it. We also know, as you say, that there is work being done on that, and so we're hoping to interface with some of that work, I think. But that's definitely an important aspect as well. I didn't talk a lot about specimen and specimens collection and vouchering here because we haven't developed that portion quite as much yet, but it is a section we're working on. So.
So any questions? Or maybe you can contact Mark later. So I will send the mails. Yeah, the
Great,
report
thank you.
to everyone. Yeah, thank you. Thank you. Thank you, Professor Mark. So yeah, so next speakers I found Davey and Adams were online. So you both of you will make the presentation?
No, just me today.
Okay, yeah.
That'd get confusing, I think.
No, thank you. Thank you. And just make a brief introduction. And Dr. Davy and Dr. Adams is from the United States Department of Agriculture. So and this afternoon I also read the papers about the fair bioheaders of reference genomes. And I found that because right now the fair principle is accepted by more and more people and more teams. Yeah, and they have some extension and some work on this part, and hope that every attenders can get some ideas from their presentation. So okay, Davey,
Absolutely.
please.
Yeah, let me
Yeah.
share my screen, set the whole thing up. I posted some links in chat, which will be useful for the attendees of this workshop. Sure. Slideshow. Okay. I think I'll start off with just a bit of an introduction of Adam and myself. So I am a computational biologist and a principal investigator for the United States Department of Agriculture Arthropod-Borne Animal Disease Research Unit in Manhattan, Kansas. Now, primarily, I study gene regulation in agriculturally relevant vector insects. By almost definition, these are non-model insects, which have their own problems. And I would love to talk about that, but today we're going to be talking about sort of my secondary focus, which is data standards and perhaps fulfilling some of the standards gaps that Mark just talked about. Adam is a software architect and a technical manager for the Reactome project, a software engineer with the Alliance of Genome Resources, including Wormbase, and of course, my co-lead. We are both co-leads of the FAIR Bioheaders project. I've posted our relevant GitHub link in chat and our recent-ish manuscript for FAIR Bioheaders reference genome, as we intend to think about this same problem in other spaces. But you can see relevant code there. Without further ado, there's a problem within reference genomes, often overlooked, which is that copying data from a central data repository onto analysis platforms, think your university or institute's high performance computing center, or even your own desktop, could induce errors in or confusion in the reference genome. So, and then oftentimes we've noticed that a reference genome won't just get shared from one data repository directly to where it's analyzed, but from one data repository to a high performance computing center to somebody's desktop and maybe around a lab. This recursive sharing can introduce various changes, sometimes due to copy errors. Large genome or large files in generally can have packet loss, which will reduce the file size. This is one of the principal reasons that people started thinking about checksums on reference genome assemblies. But we've also noticed that when these files are getting shared around, sometimes what can happen is that there'll be small annotations or small edits to the file, and so that the chain of what we'd call data integrity is lost. So thinking about this, we wanted to create a standard which could, and when I say standard, I'm using that in the loosest of terms. We're talking about a schema with associated serializations. And I would more often than not in a presentation call it a serialization and not a standard. There's a big difference. Only the schema is a standard. Everything else is a serialization, if you want to get technical about it. But what we wanted to do is create a standard that could with minimum invasiveness, a light touch, preserve data provenance from system to system or across systems. And we wanted to do this in such a way that at any point within this chain, we could validate that. So, and you could think about this in terms of perhaps, you have NCBI reference genomes. They're being sort of linked to by one resource, say like the I5K website, and then that's going to, some JBrowse instance somewhere or the Apollo instance, and we want to continue to have data integrity through that system. So there's two solutions to sort of keep data integrity. One is a machine actionable way. I already referenced it with checksums. So a checksum is just a hash, which a lossy hash of the data itself. And so you can recreate that using the same algorithm from system to system. I press a button, I check that my file is validated, it's validated, you're good to go. But another way to think about this is through data provenance and a collection of metadata about that data that would feasibly return what the file that you're looking at is, where it came from. And so FHR tries to do it both ways. To do this, we use several serializations. We use a schema, and we've written libraries that can be both used as separate tools, but then imported as Python libraries into other tools that can utilize the validation and creation that we've written. So you can essentially import it into whatever. A key component of this is the algorithm checksum. As I alluded to, it's a string representation of a document, each document a unique string, and it can tell you whether or not it's complete. So if you have data packet loss, it would tell you that, and that created a write error. Actually, sometimes what I've seen is that with large files, not to scare anyone in the chat, with large files, especially in older systems, if you're writing to a very large file, there's a data backup problem, and you'll actually miss a component. Think about this. If one place where I saw this is I was using an API, won't say whose, and I was hitting this API in 4 different ways on 4 different analyses. And so I expected the same number of returns from each of these APIs. Of course, over 1 million hits, right? And I'm hitting the API 1 million times 4, so 4 million times. Those files did not match up. So I have to go back, figure out what got missed, and then rerun that analysis. Now, the easier way to do that is, through validation checking in your API calls. You should be doing that. But again, it can happen if you're not paying attention. And checksum can also tell, irrespective of the name of the file, a checksum can also tell you whether or not that file is the same as another file. And so if you've got two genomes with two different names, say HG17 and what is it, GH, I'm not a human researcher, but they're, the same human genome it's the same thing from one to the other, but they have two different file names with two different, sort of provenances, then a checksum can tell you whether or not those documents are the same. So including that in any document recording or provenance recording is paramount. So FHR also works on recording provenance. And so it'll record things like the author of the genome, the instruments that were recorded in that, the author of the metadata, which we see as wholly separate. So somebody might write a genome, but a different team might be annotating it within an FHR serialization. And so we want to be able to, preserve that information. So we split those off. Our hope here is that in the event of catastrophic data loss, a single FHR instance somewhere would go a long way to recreating that genome assembly from scratch, or at least parts of that assembly if you needed to. It's serialized in different formats, YAML, JSON, and microdata, as well as what I'll get into, which is the preferred method, which is inclusion in the FASTA itself. And we do some really interesting things to be able to do that. And then the converter was written to validate and convert between formats. And again, and I can't stress this enough, FHR strives to be as minimal as possible. We do, we work on this idea that all things in science take effort, and we want it, you have to sort of be strategic about where you want to spend your time. And, we don't want to make a biocurator use a quarter of an FTE to, write FHR headers. And so we strive to lower that action potential as much as possible. Just, here, use this tool. It gets you most of the way. Then, all you really need to know is the information. Suddenly you've got a much more data integrity, a much more or much higher data integrity than you had before. So the main way that we hope that FHR is implemented is through the FASTA format itself. So FASTA created in the 1980s, it's been around for a while, and hasn't changed much since. You have a greater than sign, which talks about the header of a contig, a sequence, really. We use it for contigs and scaffolds, and we use it in different ways. And what you may or may not know is that in the original specification of FASTA, there actually was a comment line. And the comment lines were denoted with a semicolon. Of course, not nobody, but very few people use, if at all, use the comment line today. But some tools, I'm thinking of like the R sequencer library, use a legacy mode, which will ignore the semicolon. And so what essentially we do is we write all of the metadata in these comment lines to at least reduce the hurt that it would cause to add headers to FASTA files, making it slightly easier for an environment where you'd have these in, multiple reference genomes. Less code writing. But we also, in order to control for that, have written a tool that strips the header out and then puts it into a sidecar file if it's absolutely necessary. So it should be in the FASTA. We want it to be in the FASTA, but we understand that we live in an environment where that might be kind of a tough call, and so, or a tough thing to do. And so we've written tools that can control for that as well. Again, lowering the action potential of adoption. Yeah, so our other serializations include microdata, which is basically HTML tags, but a certain way to use them so that things like web crawlers can use them. That comes into play in our sort of next steps and future directions. Talk about that. YAML and JSON. So this minimal disruption, maximum reward is key to sort of the fair bioheaders mentality. So we want to put the header in the FASTA so it continues to exist with the FASTA in perpetuity, but we provide these other serializations to share different possible scenarios. And then we want a maximum reward. FHR records necessary metadata that could otherwise be lost through sharing the file over different systems, but and it also records the checksum within the file. So we have to be very clever about how we take that checksum out, look at the file and put it back in whenever we validate, so that we wrote a whole tool to control for that. And can be used as a unique identifier in a way for the file itself. Some of our required fields, almost exactly what you would expect. Version, the genome, synonyms for the genome names. We recognize that sometimes multiple names will get attributed to the same file. The taxon that it's looking at, we require identifiers.org taxonomy, the metadata author and assembly author, which I talked about, date created, physical samples, which is actually turned into a new field called voucher. All of your reference genomes should be vouchered somewhere physically. And if it's not physically a voucher, it should be the colony that it was from, the location that the assembly was created. That's the sequencing center, instruments, assembly software. Actually, come to think of it, everything after instrument is not a required field, but there to increase the amount of provenance in the file. We go through a lengthy debate every time somebody asks for a new field, and we do discussions on whether or not that field would really increase the usefulness of the file versus increase the sort of clutter of the header. This is not intended to be the full description that you might have somewhere in a database. It's intended to be the minimal requirements. Again, can't stress that enough. So this is what an example header would look like. You have the semicolon, and then we use a secondary character, and this we use the tilde. This denotes comments from just regular old comments from the header. And so the header can be anywhere within the file. It can also be interspersed throughout the file if so needed. The validation software will just strip all of the lines with the comments and then take the ones with the tilde. And there's a whole story about why a tilde, but then you'll have the data, which is then imported into the Python library. It could be used in further analysis if so wanted. So we designed this under the auspices of FAIR principles. Now, this is a schema and a standard, or schema is a standard, serializations or serializations, but we wanted to make sure, and so therefore it itself cannot be fair because only data can be fair. But we wanted to ensure that the standard and the serializations were increasing for the data, the ability to reach fair and the ability to reach trust will I get into. So I won't go into this now in the interest of time, but we go through and we look at findability, accessibility, interoperability, and reuse, and from the framework of using a fair bioheader. We also do that with trust, which is transparency, responsibility, user-focused sustainability, technology. We also have started embedding the ability to use fair headers into other software. As of version 1.7.9 of JBrowse, you can it will strip the FHR header out of a reference genome. Embedded server concept would be something like an information button on, say, like the BlastX website, where you can press this and it returns the fair bioheader information. As I alluded to, we're recently published. You can check that out. Our next step, or we're going to be taking our microdata serialization and sidecar. We're going to be adding that into many genome notes papers now. We'll have this big old table of all of the provenance. Our push is to standardize that provenance in conjunction with micro publications. And we're in talks with the Sanger Institute, which will tag that table with all of the provenance information, the genome notes, and then a FHR header will be able to return from that genome notes table. With that, special thanks to all of the co-authors and all of the people that we had many fruitful discussions with. I won't go through the entire list, but needless to say, a very strong, very fruitful discussions came out of all of this. With that, I'll, in the interest of time, I'll take any questions or any discussion.
Hello, David. Did you hear
Yes.
me?
Yes.
Oh, yeah. Thanks for your talk. My name is Raymond. I was working on the whole and honeybee genome assembly. And then one thing I'm not quite sure, 100%, I'm not sure whether I understand the FHR correctly, because in one slide you mentioned that if I lost my genome assembly, for example, I delete my genome assembly by accident, but if I have the FHR instance, I can use it to regenerate or recreate my genome assembly, because I have put the pipeline or how to produce my genome assembly into the FHR. I'm not quite sure whether I understand
Right.
this correctly. If I'm wrong, you can correct me.
Yeah. So the question was essentially, I alluded to being able to regenerate assembly from an FHR instance. And what I mean by that is not, regenerating the GATCs, but having the ability, if you were to lose the assembly file. So this is not computationally. This is the worst this happened, there's a power outage in several areas that have reduced the reduced computational infrastructure to, and this is obviously hopefully never going to happen, but the file is lost. But with an FHR header, I know what at least was done for that file. I know who created it, and I know where it was from. I know, what tools were used to create it. And so if I had to go back to the wet lab, do the DNA extractions again, go to the sequencing core, then, assemble it with with a certain assembly tools, software, I would be able to know some of the information that, or I would know the information of all of the steps that were taken in that, or at least be able to sort of go to the people that were involved and be able to run the experiment again, or run the like from the start. I did not try to, I don't want to imply that we're able to regenerate the contigs and the scaffolds themselves. You'd have to resequence and do all the things. When I say catastrophic, I mean catastrophic in the worst way. A very unlikely scenario, but a useful frame on which to look at. And I used to think about, standards and minimal fields. Do we really need this field if we know nothing about this file, kind of thing? I hope that begins to
I got it. Thanks. Yeah. Thank you. Thanks, David.
Okay, thank you, David. And I think we need to move to the next topic. And yeah, thank you. Thank you, Kim, for waiting so long. And Dr. Kim is acting director. We're happy to invite her to join this workshop. And the acting director of the NCBI, and used to be a chief. I get the info from the web page that it used to be the chief of the information engineering branches at NCBI. And today we'll share some biomedical data repository concept. But maybe everyone think that this concept is far from because we focus on some biodiversity. But I think even in China, I'm not sure that every countries, the plant research or some animal research, we transfer the data. We find some useful genes can be used in some biomedical research or biomedical medicines things. So I'm happy that Dr. Kim can share his ideas about this part. So go on. Yeah.
Thank you.
Thank
can
you.
You see my slides? I want to make sure that the right monitor got shared.
Yeah.
Okay.
Okay.
Thank you very much. Good day, everybody. So as Xiaofeng introduced me, I'm Kim Pruitt, Acting Director of the National Center for Biotechnology Information, otherwise known as NCBI, and we are a division of the National Library of Medicine at the National Institutes of Health. And so, let's see, I am going to be giving you a summary of a publication. I stuck it in the chat so you can read it for yourself, and there's richer information there. I'm going to be going over some concepts, some principles and properties, management best practices, touch on briefly at the end, community resources that are really about repository management. A lot of people talk about FAIR, but not a lot of people talk about best practices around managing data repositories. And so that is my focus today. So I'm going to start with the definition. And this is the work of a NIH working group that included staff that came from several different ICs at NIH. And so a data repository is defined by NIH as a system that accepts submissions of data from researchers or public health groups or others, and provides services to store, organize, validate, preserve, archive, and redistribute the data for reuse. In contrast, NIH defines a knowledge base as a system that extracts, organizes, and annotates core data sets that are managed by data repositories. And knowledge bases don't typically accept submissions, but instead focus on extracting meaningful knowledge from these other information sources. And then the terminology data resource is something that we use to refer much more broadly to any collection of data that's been organized and managed to serve a specific purpose. And so it's an umbrella term over knowledge base and data repository. And all three of these, repository, knowledge base, and resource, incorporate and manage collections of discrete related data objects or data sets. The repositories can be classified into different types, which are shown here. Domain-specific repositories accumulate a specific kind of data. So it's really as it's described. It could be image data or it could be sequence data, but the data type is specific in those kinds of repositories. Generalist repositories do not have specific types of data. They are organized around a very broad type of data, anything that you want to put into a generalist repository. We also recognize that there can be project specific, and so like a data, like there could be I5K as an example, could have project specific data management and repositories that are organized around the needs of the insect community. And then there are also institutional repositories. And so, for example, an academic organization, a university, they may set up a universal an institutional repository to manage the research outputs of the scientists who work at that university. Repositories can be further described by these different kinds of characteristics. What community do they serve? What kind of engagements do they have with the community? What level of curation do they do? How do they preserve data? What other kinds of services? And of course, what type of data do they manage? And access for repositories can be open, which is, it's free, publicly available to anybody without registration needs. It could be something that requires registration. It could be controlled access, as we see very frequently with sensitive human sort of genomic data. It could be behind a paywall. It could be a repository that has looked to a model where the community has to help support them by paying some type of fee. Sometimes it's voluntary, but sometimes it's required. Or it could be proprietary.
Sorry, Kim?
Yeah.
Sorry, Kim, I sharing.
Is there a problem?
1 minute.
1 minute.
Yeah,
okay.
you can.
Okay.
Yeah.
So the paper talks about the intersection of these items in the management of, if it's only 1 minute, I think I'm just going to end here.
Maybe you can share the screen again. Yeah, maybe you can share the screen again.
You can't see my screen?
Yeah.
Oh. I thought it was, my system told me.
Yeah, 1 minute before.
I was seeing it at that time.
Yeah, can you see my screen?
Hey, okay now.
Yes.
Okay. Yeah, so if you want me to end in 1 minute, I'd like to end here. The point is that we describe a life cycle management, a life cycle for biomedical data repositories. This is something that is like one of the novel outputs of this working group. And we also describe how these things intersect, FAIR principles, trust principles, which is about repository management, and how metrics are core to being able to assess the health of repositories, both in terms of where it is in its life cycle and how it is meeting the trust and FAIR principles. And I would then just like to invite people to read the publication, and I'm happy to talk to you later on about any of the elements that you might be interested in. Because I think I'm out of time, right, Vincent?
No, Kim, you can keep going.
Yeah.
We
Yeah, keep going.
I'm sorry.
Yes,
Yeah,
yes.
keep going. Yeah.
Use your time. Yes, so we are sending the
Yeah.
link to the people in the second session, so they're joining now to actually see the end of the
Yeah.
talk. So it's
Okay.
all good.
Yes.
Sorry.
Sorry, I just added that there is 1 minute, but we cannot see the screen. Yeah.
Oh, okay. Sorry. So the trust principles have been described in a publication by Dr. Lin et al. And these are principles for management of data repositories. And so, repositories need to be transparent. They need to have good public documentation describing their services and the data holdings that they have. Repositories hold a responsibility to the community for ensuring the authenticity and integrity of the data that they're holding. Repositories must maintain a focus on their user base. It's not, they need to make sure that data management norms and expectations of these communities are being met. And the data dissemination is serving the needs of the communities that they're supposed to be serving. Repositories should be efficient and effective in their data management and IT practices in order to be optimize their chance of sustaining services and preserving data for the long term. And of course, repositories need to keep up to date with technology. They need to be able to continue to say that the services they're providing are secure, persistent, and reliable. So following the publication of the trust data principles, the U.S. office, the White House Office of OSCP, Office of Science, Technology, and Policy, published the desirable characteristics for data repositories, and the link is here on the bottom of my slide. And NIH also published guidance around desirable characteristics for data repositories, and they're sort of summarized on this slide. There's they can be categorized into sort of 3 kind of buckets here: organizational infrastructure, and that's like the expectation is that it's free and easy access. There's the maintenance of submitter confidentiality. There's clear documentation about using the data, the retention policy, and that the organization is carrying out risk management practices. Around data management, providing identifiers for the data that's being held that are persistent, collecting and maintaining useful metadata, clear data provenance, providing QA, QC, curation services on the data to ensure that the data is of high quality to support reuse. All of these are to support downstream, the fairness of the data, right? And so data repositories have these requirements in order to help support the fairness of the data. It all works together. So here, I did want to give you a little bit of an introduction to the repository lifecycle. And so assessing both the fairness of the data and the trustworthiness of the data repositories occurs within the context of lifecycle concepts. So data set life cycles have been proposed and described by other groups, and I'm not going to talk about that here today. But we found very few descriptions of biomedical data repository life cycles. And so we adapted existing, the standard product life cycle description for the health sciences. And that's what's shown here on this screen. And so the introductory phase involves the initial development and establishment of a repository, which is perhaps being generated to meet new scientific needs. The first introduction may really be a minimum viable product with only a partial set of features. User feedback is actively gathered during this phase. User testing is solicited. And the feedback from those users and the community informs on the growth phase, where additional features are added, so that it's moving then from a minimal viable product into a operational product during that growth phase. The addition of features is prioritized based on input and feedback from the community about what their needs are in a product data repository of this type. And during this phase, the repository sees an increase in usage and community adoption. And generally, you know, this is occurring along a time scale, but the length of time in each phase is going to be variable depending on the particular thing that's being developed and introduced. Repositories during the growth phase focus on increasing the data holdings and improving the user experience through enhanced accessibility and other kind of approaches. At some point, the growth phase sort of, you know, tailors out into a maturity phase. And so at the maturity phase, this data repository has achieved significant user adoption, and features will continue to be optimized, but the rate of change and the rate of user growth like that slope has decreased into a flatter part of the curve. Maturity phase can last for years or decades, or you know, so the length of time that something may be in the maturity phase can be quite long or variable even. And repositories may eventually reach a phase that we call decline or disruption. And so this can occur for many different kinds of reasons. And so it could be that the type of data is no longer relevant. And so I'll give you an example. Here at NCBI, we had a resource called Homologene that was based on clustering of EST data. I'm sorry, I'm saying the wrong thing. Unigene, that was based on clustering of EST data. And as you know, early in the genome project, EST data was a critical approach to be able to get sort of a crude assessment of how many genes there could be. And so the sequencing technology evolved, and we no longer use ESTs really heavily. And the resource Unigene was no longer really needed. We had well-annotated genomes. And so Unigene was no longer really relevant to the user community. So that's a decrease based on relevance. The decline can also be based on technology changes. So technology's moved on, and the users go to the repository that hasn't kept up with the technology. And they're going to this repository, and it's like, this is clunky, it's hard to use. You know, I don't want to come here anymore. I want to go to some other repository that has similar information, that's a much more intuitive user interface. And so that technology change then results in the fact that the repository didn't keep up with technology changes results in a user community that no longer goes there. And so literally the number of users accessing that starts to go down. And the other reason that there could be a decline or disruption could be a loss of funding or a decrease of funding below the amount needed to sustain the repository, or departure of critical staff. You know, the PI leaves and closes his lab, and there's nobody left to take care of the repository. So this decline phase is a complex phase. But at some point, the decline reaches, or the disruption reaches a point that we call the decision point. And a decision point is really when the repository managers or the organization and institution needs to assess that repository's future trajectory. Does it still meet a scientific need? Is there a way to stabilize the funding? Do we have stability in staffing to manage that repository, and they need to actually make a decision whether or not to sunset that repository or to reinvest in it, such as to modernize the technology, modernize the user interface, change things to better meet the current user needs. And so this repository lifecycle model, I think, works very well for NIH repositories, and I think it probably will work well for other biomedical repositories. And of course, the last important piece of this is metrics. And so metrics are key. And so repository managers are utilizing metrics in they're utilizing many different kinds of metrics for several different purposes. And so they're understanding usage trends, they're understanding user behavior. And so, like, is usage going up or is it going down? Are people staying on the pages for a long time? Are they doing multiple queries? How many users are going to the page? What are they clicking on the page? And so this user behavior category is like one of the core categories of metrics, right? How many times are they downloading the information? Where are they coming from? How global is your user base? This also, metrics are also used to assess or to try to assess, the level of contribution and impact of the data that's being held. And so if you're looking at something, you might be asking, well, what are the number of projects or studies that repository is receiving? And what are the number of cases or subjects in those projects or studies that the repository is managing data for? And everybody also tries to do some level of bibliometrics, which is difficult. It's the best we have right now for assessing scientific impact of repository, is by seeing how many citations that repository gets in papers. But often repositories are sort of taken for granted and they aren't cited in publications, and so it's not a satisfactory measure for scientific impact of that repository. And this is an area where much more research is needed. There are gaps here in being able to really understand the impact that repositories make and the holdings of the data they're holding. What impact does that repository have on society, on public health, on the advancement of knowledge, right? And so more metrics are needed in that area. Metrics are also used to understand repository operations. We're looking at what are our storage costs? What's the cost per data set? How fast are we processing submissions? How much does it cost per download? What can we do to make this more efficient? And making that more efficient makes the repository more sustainable. And there are numerous community resources in this space, and just a few of them are mentioned here. And these communities are working on elaborating best practices, elaborating on standards. And so standards around how to manage data or standards around data formats, right? That can help make repositories more sustainable and more efficient. And standards about how do you know if your repository, is meeting the desirable characteristics, is meeting the trust principles. I'll highlight Core Trust Seal. It's a certification. You can go to Core Trust Seal and actually have your repository certified as a way of publicly communicating, that your repository is meeting the trust principles. Make Data Count is an interesting community. They are working on elaborating on different kinds of metrics around assessing data and assessing impact. And Research Data Alliance has also been very active in that space as a community formed organization. And I'm almost finished. And so, here's my sort of my bottom line, right? Repositories, I feel, repositories in their operations, their trustworthy operations, are essential for scientific research. They're essential for both preserving the knowledge that comes out of scientific research, redistributing that knowledge so that others can reuse it, so that we can build upon You know, we're constantly building upon what we already know by new research endeavors. The repository landscape has been, is, and will be very diverse and continually evolving. Best operational practices are now emerging for trustworthiness, lifecycle management, and assessment metrics. And community efforts on all of these things, the information that's relevant to repository management continues to mature and evolve. And so I think this is a very active space. I'm really excited to see some of these, you know, some of these principles and best practices starting to come out and be adopted by other groups. And I'm going to end there. And I'm happy to talk to anybody further about this later on. You know, we're over time and we should move on to the next talk.
Okay, thank you. I really learned a lot from your presentation because, you know, National GeneBank, I also have a teams focus on data repository, but never stores. I think we never deep thinking about the data repository, the life cycle. There is a model of life cycle because I think maybe I think in the future you can make another presentation EBP because I think most of the projects in EBP, they have a repository, they have database, but they have no framework or concept they how to manage their database. Because right now in the world there is many bioinformatic database, but 3 years or 5 years they close their database. Yeah.
Yeah.
Great.
Yeah.
Thank you. Thank you, Kim. Yeah.
You're welcome.
Any questions? Yeah.
Yeah, so I guess we may benefit from asking questions
Maybe
later. I don't know if Kim will be able to be in the
Yeah.
next session, but either way, there is the Discord chat
Yeah.
for those that are
Mhm.
attending, and maybe we can start a conversation there.
Mm.
Oh,
Yeah, I'm
Okay.
taking this is such an important topic to discuss, and it's really important that people are aware. Yeah, I just keep thinking about
really.
gold.
Yeah. Yeah. Also, I was thinking about our platform in National GeneBank. Yeah.
Okay, so
Right.
with that, maybe we should just move to the next Zoom link to join all the other
Yeah. Yeah. I also
presenters.
put a Zoom link in the chat. I hope that everyone can also join this link. See you later.
Yeah,
Thank you. Yeah.
thank you. See you later.
See you later. Thank you, Kim. Thank you.